A short history of telomere research. In humans, the thing is that as we mature, our telomeres slowly wear down. So the question has always been, did that matter? Well, more and more, it seems like it matters. Elizabeth Blackburn. In the late 1800s, the German scientist August Weismann put forward an intriguing theory about the biological basis of aging. His hypothesis suggested that aging did not occur by the waste of single cells, but rather by the inability of cells to renew tissue because their capacity for cell division was not everlasting, but finite. In other words, he thought the aging process stemmed not from deterioration within individual cells, but rather from the limited capacity of cells to divide and regenerate tissues. At the time, technology had not advanced sufficiently to test this revolutionary concept experimentally. Shortly before Weismann's death, his theory faced a significant challenge when Alexis Carrel published findings suggesting that mammalian cells possessed unlimited replication potential. This view dominated scientific thinking throughout the early 1900s. Carrel, who had achieved prominence through his groundbreaking work on vascular suturing techniques, earning him the 1912 Nobel Prize in Physiology or Medicine, even graced the cover of Time magazine in 1938. During this period, he collaborated with Charles Lindbergh, yes, the same Charles Lindbergh who became famous for completing the first nonstop flight from New York City to Paris, to develop the first perfusion pump. Carrel's experimental work centered on chicken heart cells that continued dividing in culture for an extraordinary duration, outliving Carrel himself. The responsibility for maintaining these cells transferred to his colleague Albert Ebeling in 1912, and they continued proliferating until 1946, surviving for 34 years, remarkably longer than a chicken's typical 5 to 10 year lifespan. The paradigm of cellular immortality remained unchallenged until 1961, when Leonard Hayflick demonstrated that human cells could only undergo a finite number of divisions in culture. This maximum number of divisions became known as the Hayflick limit. Subsequent research confirmed that human cells typically divide between 40 and 60 times before entering a non-dividing state called senescence. Scientists observed similar limitations across various species, including mice, horses, and tortoises, with the number of possible divisions correlating to each species' lifespan. They also found that cells from older donors reached senescence after fewer divisions. The reason for Carrel's contradictory results remains uncertain, though some researchers speculate that new cells may have inadvertently contaminated his cultures when fresh medium was added. The early 1970s saw Russian theoretical biologist Alexei Olovnikov synthesize Hayflick's findings with contemporary knowledge about telomeres to propose a connection between these chromosome structures, aging, and cancer. The story of telomere research had begun decades earlier, in the 1930s, when Barbara McClintock identified distinctive structures at chromosome ends while studying maize. Further investigations by McClintock and Hermann Muller revealed that these natural chromosome ends served a protective function, behaving differently from broken chromosome fragments produced by X-rays or during meiosis. Muller coined the term telomere, combining Greek words for end, telos, and part, meros. Following Watson and Crick's 1953 description of DNA structure and Arthur Kornberg's characterization of DNA polymerase, James Watson identified a fundamental problem. DNA polymerase could not completely replicate DNA strands, resulting in progressive shortening of chromosome ends with each replication. This observation suggested that telomeres must gradually shorten unless cells possessed some mechanism for maintaining their length. This mechanism was eventually identified as the enzyme telomerase, discovered in the 1980s by Elizabeth H. Blackburn, Carol W. Greider, and Jack Szostak, work that earned them the 2009 Nobel Prize in Physiology or Medicine. Though humans possess the cellular machinery for telomere maintenance, most human cells don't express telomerase. Research in 1986 confirmed that human telomeres indeed shorten with age. However, certain cell types maintain high telomerase expression, germline cells and cancer cells. This makes sense as both cell types exhibit immortal characteristics, replicating without ever reaching a state of senescence. Stem cells also express telomerase, though typically not at levels sufficient to prevent age-related telomere shortening. The germ line, the reproductive line of cells, has been replicating for roughly 4 billion years of Earth's biological history, maintaining continuity through countless generations of replication and mutation to the present day. Cancer cells demonstrate similar immortality. The HeLa cell line, derived from the tumor of Henrietta Lacks in 1951, continues proliferating in laboratories worldwide despite her death that same year. Research in 1994 revealed telomerase expression in most cancers, with subsequent studies showing significant telomerase activity in 85 to 95% of human cancers. In a fitting twist of scientific irony, the first non-cancerous human cells to achieve immortality through artificial telomerase introduction came from Dr. Hayflick himself, the very scientist who had demonstrated that human cells would senesce after a limited number of divisions. This achievement proved that telomerase alone could grant human cells unlimited replicative potential.